Hello, my name is Caitlin, and this is modeling gene switches of Pitx1 and comparing and contrasting prokaryotic and eukaryotic gene regulation. This first model is of the marine stickleback pelvis tissue with the transcription on. This model shows the pelvic switch connected to the pelvic switch activator. This pelvic switch activator is connected to the mediators, the general transcription factors, and the RNA polymerase. Once these are all connected, they create a bent shape in the DNA, and the RNA polymerase is then connected to the promoter, which then cause the Pitx1 to express the pelvic tissue. This next model is of the marine stickleback jaw tissue. This model is similar to the first one with the pelvic switch. However, this one shows the jaw switch connected to the jaw activator. This then allows for the jaw tissues to be expressed. This model is of the freshwater stickleback jaw tissue transcription on. This model is different from the first two as it does not have, or it has an inactive pelvic switch activator. But again, it does show the jaw switch connected to the jaw activator, allowing for the RNA polymerase to begin expressing the jaw tissues. This model is of the freshwater stickleback jaw tissues with the transcription off. This model shows the jaw switch and the pelvic switch are inactive, and the jaw switch activator is unable to connect to the jaw switch. This doesn't allow for the jaw switch or the Pitx1 gene to express the jaw tissues. This model is of the TRP operon. It contains the promoter, the operator, and the structural genes. The operator is usually bound to a repressor. However, this repressor can be bound to an inducer. This inducer changes the conformational shape of the repressor and opens up the operator. When this operator is open, the RNA polymerase can connect to the promoter. This allows for the RNA polymerase to begin transcription of the structural genes. When transcription occurs with the structural genes, they create mRNA. This mRNA is then translated into proteins, and this eventually leads to the synthesis of proteins and tissues. Next is prokaryotic versus eukaryotic gene regulation. Both eukaryotic and prokaryotic cells regulate gene expression based on internal and external stimuli. They both use RNA polymerase to go through the process of transcription. Prokaryotic cells regulate gene expression through controlling transcription. This is solely through operons, seen in the model we just reviewed, where the inducer binds to the repressor, opening up the operator and allowing the RNA polymerase to transcribe genes. When there's no inducer, the repressor is bound to the operator, and the RNA polymerase is unable to transcribe those structural genes. In eukaryotic cells, the process of gene regulation is more complex. Transcription factors and mediators are bound together. Which are then bound to activators. This causes DNA to be in a bent shape. Again, eukaryotic cells use RNA to transcribe genes.